Now, here's the thing. We can actually look at the code in your DNA and make a hypothesis as to what the amino acid sequence should be to the corresponding protein. And then we can do all sorts of research from there, looking at those amino acid sequences, predicting how they're going to fold up in your body, and then what kind of job they might do. So, how does this whole thing work? Well, first and foremost, I have to remember that I'm reading this as a triplet code. So every 3 nucleotides ultimately corresponds to a codon, which then translates into a particular amino acid. The other thing I have to remember when doing this transcription and translation from the code standpoint is that those T's replace U's in RNA. So there are no T's in RNA molecules, only U's, but the rest of my base pairing rules apply. So instead of A to T, it's going to be A to U, but C to G will stay the same. So in your notes, you should have this DNA code. And I like to go through and draw lines every 3 bases just to make this a little easier for me to kind of navigate to keep organized. So I suggest you do something of that nature. I've also had kids who will go through and underline every 3 bases or go through and highlight in different colors every 3 bases. Whatever helps. It's just going to really come down to needing to be organized. So figuring out whatever method works for you, go for it. All right. So if I'm going to take a look at doing the transcription piece, I'm going to go from DNA to mRNA. And that means that my mRNA sequence has to be complementary to the DNA one. So what's its opposite? So if I look here, if I've got ATG as my first codon in DNA, that means I need to know what its opposite is in terms of a codon in mRNA. So A becomes a U because there's no T's, right? T becomes an A, G becomes a C. And then I can work my way down that DNA sequence to make the complementary mRNA. And then it should look something like this. Okay, so 3 A's would become 3 U's, 2 A's, 2 U's, a C to a G, so on and so forth. Now, I can also take a look at this mRNA code and predict the tRNA sequence that would need to be the anticodons to that. So once again, they are complementary to each other. So if I have a U here, then that means I'd have an A in tRNA. If I have an A here, then I'd have a U. If I have a C here, I'd have a G, so on and so forth. So my tRNA sequence should look something like that. And if you notice, and if you've done this correctly, so this is a great self-check, my tRNA sequence should be the same as my DNA with one exception, right? Because there are no T's in RNA, I've U's instead. So this should be the same as my DNA, except U's instead of T's. Okay, so hopefully that's not too bad. The last piece is actually figuring out the amino acid sequence that this all corresponds to, and that's where this table comes in. This is a codon table. Now, here's the thing you have to remember when doing this. This entire table is built off of your mRNA sequence. So when I'm going to create this mRNA sequence, I have to come back to my messenger RNA. Otherwise, this table is going to get you the wrong answers if you're looking at the wrong sequence. So if I look at my messenger RNA, all right, here's my first codon, UAC. Now, if I look at the way this table is set up, my first codon corresponds to each row, the first base in the codon. So the U is the first one I'm looking for. So if I look, every codon that starts with a U is in this row. Every codon that starts with a C is in this row. Every codon that starts with an A is in this row, and then every codon that starts with a G is here. So because I've got a U first, I'm going to focus on this very first row. Everything else below it is irrelevant to me, at least in this particular situation. So I'm going to focus on this top row because my first base is a U. Then I can go down the columns. That's going to correspond to the second base. So all my U's are in the first column, C's in the second, A's in the third, G's in the fourth column, based on the second letter in the codon. So go back to my messenger RNA. I've got a U as my first, A as my second. So I'm in this row, and now I know I'm in this column, so I'm somewhere in this box. Then I could look through the rows again in this direction. But personally, because by the time I've gotten that far, I've only got 4 bases to look at, or for 4 options, I should say, to look at. I kind of just go with it and look in the box. But if using this extra row is helpful to you, then please, by all means, go for it. If I look at this, I've got a U, an A, which is what's gotten me to this box in the first place. Now I've got a C. So here's my UAC. Okay, and so that UAC corresponds to this amino acid right here, TYR. Okay, and so that's the first amino acid in this particular sequence. And then I would go through my next codon, 3 U's, so U gets me back into this row. Second U gets me into this box. Here's my UUU, which is the PHE or the phenylalanine. Go down to the next one, 2 U's and a G. All right, gets me right here to leucine, so on and so forth. And so you could work your way through getting the rest of those amino acids. Something else I want to point out here, while we're on this topic of reading RNA, first and foremost, I've given you just some snippet in the middle of a protein sequence. If I were to truly give you the full code, your first mRNA codon always has to be AUG, which means the first amino acid in your sequence always has to be MET. All right, I haven't given you the beginning of the protein code, which is why you don't see that. But if I did, that's where it would start, and that's what you would see in every protein that your cells produce, because that's how it knows that that's the start. Same thing here, I didn't give you the end either. We would have to keep reading, your cell would keep reading, your ribosome would keep reading until it hit one of these 3 stop codons, and then everything would just fall apart and it would stop. So AUG is always where you start if you have the whole sequence. Stop is where you would end, once again, if you have the full sequence. You don't here, but you may see that in some practice worksheets outside of the notes themselves, so just be aware of that.